Now, let's talk class 2. Class 2 is going to share nothing with class 1 and its class 1 loading. Completely different proteins because we're talking about a completely different compartment. Okay, nothing here cytoplasmic, nothing here ubiquitinated, no TAPs, none of that. So what we see here in the far left of the figure is something outside the cell. Triggers the cell to take it up. Phagocytosis, pinocytosis, whatever. Goes into an early endosome, which has a near neutral pH. You know, the pH of whatever is the space, that extracellular space around our macrophage, our dendritic cell, whatever we're talking about here. As that endosome begins fusing with lysosomes, we see the pH drops. It becomes highly acidic, which means that proteins on the surface of that bacteria or whatever, or that protein we just took up, is going to start to denature. Going to lose quaternary, tertiary structure. Enzymes are going to go through and start randomly cutting. Some of them randomly cut every so often. Some of them go through and cut every tyrosine residue they see. So you get random lengths. This phagolysosome, or an endolysosome, depending upon which textbooks you read, will then fuse with some other vesicles. These vesicles that are from the ER will have MHC class 2 already in them. And then, just like we talked about with class 1, random insertion and bouncing out of these peptides, these epitopes, until something is able to get in and stay in stably. Now, of course, it's not going to be that easy, and we'll see why. Because our MHC class 2 is also not stable without an epitope in it. So, as it is synthesized in the rough endoplasmic reticulum, you see that the yellow highlighted areas, which are the MHC class 2, need to be stabilized by what is referred to as the invariant chain, II. II stabilizes newly synthesized MHC class 2. II comes as a trimer, as you see there. It stabilizes 3 MHC class 2 molecules. Over time, it will be degraded. As, you know, this trimer of the invariant chain and its associated class 2s bleb off in a vesicle away from the ER. It will become acidified, either as it's sitting waiting or when it binds to that endolysosome or the phagolysosome I talked about on the previous slide. The invariant chain will then be cleaved. All the green areas you see there will be cleaved away, leaving the red areas, known as that fragment is known as a clip. Clip is a temporary stabilization step. Okay? It is not interacting with the groove. It's not interacting with the MHC class 2 structure in a stable way. It is a very short-term workaround. After the class 2s, all 3 of them, have been transported to an acidified vesicle. As I said, the invariant chain, II, is going to be cleaved. That's the green areas. Here in this figure, the green is the invariant chain that's going to be removed, leaving that purple fragment. Clip. Clip is a loose association, okay? It is not interacting, it is not completely stabilizing. It will easily dissociate to become replaced by an incoming peptide. Which should be one of our just digested, just, broken down, enzymatically broken down peptide fragments.
The two chains of the MHC class II molecule assemble in the endoplasmic reticulum, the ER, with a third chain, the invariant chain, shown here in green. A portion of the invariant chain binds to the peptide-binding groove of the MHC class II molecule and prevents peptides or unfolded proteins present in the ER from binding. The invariant chain also guides the transport of the class II molecule out of the ER and through the Golgi apparatus into a vesicle that eventually becomes part of the endocytic pathway, by which pathogens and foreign proteins are taken into the cell. Progressive acidification of this endocytic vesicle activates proteases that cleave the invariant chain in two places, leaving a small peptide, the class II associated invariant chain peptide, or CLIP, bound to the class II molecule. Engulfed pathogens or their proteins are also degraded by acid-activated proteases into peptides, but these cannot immediately bind to the class II molecule because the CLIP peptide still occupies the peptide-binding groove. The removal of the CLIP peptide is the function of a specialized MHC class II-like molecule, HLA-DM, which is also present in these vesicles. It functions as a catalyst, coordinating both the release of the CLIP peptide from class II molecules and the binding of pathogen-derived peptides. The MHC class II peptide complex is then transported to the cell surface, where it can be recognized by the antigen receptors of CD4 T cells.
So, let's talk about this loading and unloading of CLIP and our peptide du jour. Far left panel at the bottom there, you see our class II molecule is being stabilized by an invariant chain. Here they're only showing one. Keep in mind, remember, it's going to be a triplex, or could be. As it blebs out in a vesicle, that vesicle will acidify as it binds to other acidic vesicles. The invariant chain will be degraded, leaving just CLIP in MHC class II. Now, third from the left, you see something is being phagocytized. Here it's just showing a protein. It could be a protein on the surface of a bacteria, parasite, whatever. Whole bacteria could be phagocytized. The parasite could be. It doesn't matter. It's going to be degraded in an acidified phagosome. You know, that lysosome-phagosome fusion is going to acidify, break it down. Proteases are going to break it down into chunks, polypeptide chunks. And then that vesicle is going to fuse with a vesicle that's containing MHC class II. What's also in that vesicle containing the MHC class II that's holding CLIP is the HLA-DM. HLA-DM in a highly acidified vesicle will associate with MHC class II. MHC class II that is stabilized by CLIP. If you've ever had me for another biology course, you've heard me say structure dictates function. Here, HLA-DM can only associate with a slightly stabilized class II molecule that has CLIP. Once CLIP has been released, HLA-DM disassociates. Okay? Disassociates and allows for an epitope to load into the cleft of MHC class II, stabilizing MHC class II on its way to be expressed on the plasma membrane. Now, a lot of what we've been talking about here has been, or I've been talking about, has been in the, This is what happens if it's a bacteria. This is what happens in a virus. Sometimes, though, some of what we see virally or bacterially, some other cell, doesn't always go to plan. For a viral epitope to be loaded into class 1, virus has to infect the cell. Virus infects the cell, gets in, is in the cytoplasm, random ubiquitation, tap and all of that, loads into class 1, goes to the surface. Well, sometimes viruses get phagocytized, which means they're going to be digested down, they're going to displace CLIP in MHC class 2, which will then be presented on to CD4 cells. CD4 cells will then take that and then present it to T cells. Or sometimes something gets phagocytized and some of the antigens are able to cross the phagolysosome membrane. Maybe they have hydrophobic portions. It means they'll insert into the membrane. Part of it gets cleaved off then. So you now have foreign antigens, foreign polypeptides in the cytoplasm. So, ubiquitation, broken down by the proteasome, sent to TAP, loaded into the ER, put into MHC class 1, presents to CD8 cells. CD8 cells will recognize that this cell has something in its cytoplasm that maybe it shouldn't, and it either tell that cell to either up its game, activate some other mechanisms for killing whatever it's phagocytized, or tell that cell to kill itself, apoptosis, thus trapping whatever that non-self thing that it phagocytized inside of it for later removal. So these are just two ways that all the rules that I've spent the last 12 minutes lecturing on can get around.